Topic 5.4 Non-Mendelian Genetics. So, non-Mendelian genetics basically refers to any inheritance patterns that do not follow Mendel's laws and they just don't segregate or they don't follow the ratios that he provided. And I'll just walk through a couple of those examples. So, the first example is codominance, which is basically when both alleles in the heterozygous genotype affect the phenotype in separate but visible ways, and it's composed of two dominant alleles. The genotypic ratio for this is a 1 to 2 to 1, and the phenotypic ratio is the same where it's 1 to 2 to 1. So the picture basically shows that one cow is fully brown where the other one's fully like tan or white, and when they are crossed over, it has both the brown and white because they're both dominant alleles. Another example is incomplete dominance, which is a pattern of inheritance in which the phenotype of the heterozygote is somewhere between the two parental phenotypes. So in this example, it would be between the red and white, it would become pink, and the genotypic and phenotypic ratio is 1 to 2 to 1. So next example is multiple alleles, and that's when there are 3 or more alleles for a single character, and that results in more than 3 phenotypes. And an example would be with blood types where they have multiple alleles. And the genotypic and phenotypic ratio is 1 to 1 to 1 to 1. Then we have polygenic traits, which is when there's an additive effect of 2 or more genes on a single phenotype. And an example of this would be skin color, because since there's 2 or more genes that affect that one trait, so that's how we get our skin colors. And then with polygenic inheritance problems, to find the number of phenotypes, you just do to find the number of alleles, you do number of phenotypes minus 1, or you do the opposite where you do number of alleles plus 1 to find number of phenotypes. Some other terms are pleiotropy, which is when one gene has multiple effects on phenotype, and an example of this would be like sickle cell anemia, because the beta globin gene has a mutation. This is because this can lead to pleiotropy with the eyes, liver, and heart. It can lead to blindness, liver failure, and then it can actually also lead to a heart attack. And epistasis is when the alleles of one gene may mask or conceal the alleles of another gene. So one locus affects the expression of another gene located somewhere else. And there's also a ratio for this, which is 9 to 3 to 4. Okay, sex-linked inheritance. So just to start off, sex-linked inheritance is when a character that is controlled by a gene on the sex chromosome, so pair number 23. And then to walk through this, basically sex-linked inheritance can happen on either the X or Y chromosome, but the X-linked pattern is more common, and plus the Y one is boring, as you've told us before. So X-linked genes have different inheritance patterns that the gene on the non-sex chromosomes, aka the autosomes, and this is because these genes are present in different copy numbers in males and females. So with this photo, we can see the fly example where 2 flies are crossed. The male has red eyes and the female has white eyes. And if their inheritance wasn't sex-linked, then it would be autosomal, meaning that all the offsprings would have red eyes because it is the dominant trait. But it is sex-linked because the mother has the recessive phenotype and all the males have received the X chromosome from their mother and they all have the mutation, so that means that is sex-linked. Also, all the females ended up with red eyes because they received the 2 X's. One is dominant from their father, and that conceals the recessive trait from their mother. Also, when following sex-linked genetics, diseases are more common in men than women because men have an X and a Y while women have 2 X's, so women can become carriers or just conceal the recessive trait. Well, men can't do that because they have an X and a Y. And this is just like an example of what I just explained, just a bit more visual. And then we have linked genes, which is when genes are located on the same chromosomes that tend to be inherited together. So linked genes, they don't assort independently because of their close proximity, and they tend to pass as a unit to the gametes. So again, with the flies, recombinant alleles, I feel like I should just define it beforehand, is when there's a combination of the phenotype not present in the parent, so it's basically like a mix. And then so on the left, it shows Mendel's prediction, which is a 25% for each phenotype, and this usually occurs when genes assort independently, and that causes equal probability of offsprings that have the parental or recombinant alleles. But on the right, it shows like the actual percentages of the parental and recombinant alleles, and it was shown that when genes are closer together on a chromosome, there's a higher probability of containing the parental genotypes. And so basically this shows how this doesn't follow Mendel's laws, and these are linked genes. And another visual aid, so when genes are closer, they're most likely linked, but when they're further apart, they're not linked, or if they're not even on the same chromosome, then they're not linked. And then recombination frequency, so this is used to construct a linkage map, which basically shows the ordered list of the gene loci on a particular chromosome, and you can see the linkage map in the bottom right corner, and you can use the formula in the top left corner to find the recombination frequency. So you just take the number of recombinants over total offsprings and multiply it by 0. If the number that you calculated is closer to 0, that means that genes are closer together. But if they're further apart, that means they're not linked. Also, the recombination frequency gives an estimate, not an exact distance. So the highest recombination frequency is 50%, which means that genes aren't linked, and lower means that they are linked. And it determines the order of the genes on a chromosome, which, as I said before, it can be used for the linkage map. Also, 1% for the recombination frequency is equal to 1 map unit. And then I'll also talk about topic 5.5, which is environmental effects on phenotype. So environment cannot change genotypes, but it can change how those genes are expressed. So an example would be soil pH, which can affect flower colors, or in humans, height and weight can be influenced by availability of nutrients. And basically a way we can like test this idea where the environment can only change how our genes are expressed is by taking individuals with the same genotype and just putting them in different environmental situations and factors and see how it affects their phenotypes. And that's it.